We will be writing an essay on acid-base balance. It was asked in second MB receipt of 2022. Now also, I'll be answering another essay that says write an essay on acidification of urine. Now for acidification of urine, that essay will entail that you'll be writing only on the things I will discuss under buffering. Because in buffering urine, it means we are acidifying urine. We are increasing the hydrogen content in urine. So these are things I want you to have in mind as we answer. So another past question has been asked where they discussed acidification of urine. So that one entirely, what the question requires from you is to discuss on buffering. Alright? So let's begin with our essay now. Our essay on acid-base balance. I'm going to introduce, and then I'm going to tell you about the response in acidosis using this outline, and then reabsorption of HCO3 minus, then buffering, response in alkalosis. I will tell you some of the major conditions, and then what eventually happens. Now, what becomes of my introduction? In my introduction, I should be able to define what is acidosis and what is alkalosis. Alkalosis, also called an increase in base levels. So acidosis is considered as a physiological state in the body, as a state in the body when there is increase in levels of hydrogen ions or a decrease in levels of bicarbonate ions. So someone is in acidosis when there is an increase in hydrogen ions or a decrease in bicarbonate ion. While someone is in alkalosis when they have a decrease in hydrogen ion or an increase in bicarbonate ion. So please take note for this in your introduction. Find them and say that in acidosis, hydrogen ions increases while bicarbonate ions decreases. While in alkalosis, hydrogen ions decreases while bicarbonate ions increases. Now for the response in acidosis, I'm going to begin by saying this. For the response in acidosis, now let's look at it and think about it together. We'll think about it together before I start answering the essay. Let's think about it together. Now what happens in acidosis? Think of it. In acidosis, you have much hydrogen ion and less bicarbonate ion. So what can you do to bring back these levels of bicarbonate towards normal? You can be able to reabsorb all bicarbonate. Try to reabsorb all the bicarbonates. Yes. And in reabsorbing bicarbonates, we can either reabsorb them at the proximal convoluted tubule, or we can reabsorb them at the distal convoluted tubule. Distal tubule. Now, at the proximal tubule, we are going to be using secondary active transport. Secondary active transport. And these accounts for about 80 to 90% of reabsorption of HCO3 minus. This is just an overview. I've not started discussing them in detail. While for distal, primary active transport, that accounts for about 10 to 20%. Now let's begin with our story. So we have said that in acidosis, we had increase in H plus and increase in HCO3 minus. Since there is a fall in HCO3 minus, to bring it up, reabsorb HCO3, and as reabsorption of HCO3 minus occurs, it takes that level ultimately towards normal. Now what do you do to H plus that is very high during acidosis? You can remove it from the body. It is lost, it is excreted. And when we excrete hydrogen ion, it joins to some substances, and that is why we say we buffer urine. In buffering urine, it means that in urine we attach it to some substances and allow it to be removed from the body. And for buffering of urine, we are going to have two mechanisms, either using the phosphate buffer or the ammonium buffer. Either we use the phosphate buffer or we use the ammonium buffer. Now for the ammonium buffer and the phosphate, ammonium buffer should be occurring in the proximal tubule. The phosphate buffer should be occurring, the phosphate, sorry, for the proximal, while the ammonium buffer for the distal tubule. I'll discuss each of them in detail. Now let's begin. We have said acidosis, there is an increase in amount of H plus. To bring it down, remove H plus from the body, that means buffer urine. While HCl3 minus is very low in acidosis, to bring it up, you should be able to reabsorb HCl3 minus. Now, what about the response in alkalosis? Now, for the response in alkalosis, as we have said, in alkalosis, there is a fall in hydrogen ion and there is a rise in HCl3 minus. So what can you do to bring down HCl3 minus towards normal? You can be able to freely filter it and then excrete. So ultimately, that is why I wrote here, I will tell you the conditions and then you will now remember that the only thing that happens in alkalosis is freely filtration of HCl3 minus, freely losing HCl3 minus from the body. So now I'll be discussing, so that means I'm going to draw 4 diagrams. A diagram to illustrate secondary active transport for the reabsorption of HCl3, a diagram to illustrate primary active transport for reabsorption of HCl3, a diagram to illustrate phosphate buffer, and a diagram to illustrate ammonium buffer. So, and all of that is in response to acidosis, and then response to alkalosis only to discuss the mechanism, and that is all for our essay. I should be able to recap something, that if you are writing an essay on acidification of urine, what that question means is that talk only on buffering urine, talk only on phosphate buffer, and only on ammonium buffer, and that is all. So let's begin. So I'm going to discuss this one, and then I will discuss this, and then we conclude with this. Now, for the first one here, before I do that, I should show you what happens normally. This is the glomerulus, this is the Bowman's capsule, and then we have our glomerulus here, and then afferent and efferent arterioles. Now, on the other side, we have our proximal tubule like this. Let me use a proximal tubule to explain what I want to draw there. Now, for every part of the nephron, there is a midline lumen. There's a lumen. And then there are specialized cells like this, specialized cells. Now, these cells are what we call renal cells. There's a lumen, there are renal cells, and then outside you are going to see probably the blood vessels. So there's a blood vessel outside, then cells, and then the lumen. This is what I'm trying to draw for you here. That if you take a cross section at this point like this, you are going to see the luminal aspects. Number one, let's say this is the lumen. Number two, what are you going to also see? You are going to see cells. These are cells. And then number three, you are going to see the interstitial space. This is interstitial space for blood vessels. So renal interstitial space. Interstitial space. Now, interestingly again, okay? So on this side also, to the lumen, cells, and then the space. So let me remove this diagram now because you understand what happens. So watch what happens at this side. I'm going to be discussing number one, that is reabsorption of HCO3 minus using secondary active transport. That's the heading. Remember, the major heading is response in acidosis. Then under the response of acidosis, you now write reabsorption of HCO3 minus. Then further under, reabsorption of HCO3 minus using secondary active transport. Now, you first of all draw a large diagram, and then you now put out the important points under it, and that is it. So let me explain to you how the diagram came about. Now, on this side here, secondary active transport means that the energy that is gotten at the basolateral side is what is held to couple transport at the apical side. The apical side is the side that always faces towards the lumen. Basal tract is the side that faces away from the lumen. So, what happens at this side here is that potassium is being restored into the cell, potassium, while sodium leaves the cell. Now, if that happens, using ATP anyway, what do you think happens inside the lumen? The lumen, as you keep removing sodium, the lumen is devoid of sodium. So sodium must enter here. And in sodium entering the cell to replenish its stores, hydrogen goes out from here. Where did this sodium come from? Actually, from filtration, we have sodium plus HCO3 minus. So we have filtered sodium and HCO3 minus. And then it is coming down. What I'm talking about, what do I mean by filter? I'm saying that if I show you the lumen like this, these glomeruli are here, like this. And then this is the lumen. So what I'm trying to tell you is that it has entered here. Remember, there are cells, 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 cells. So fluid has been filtered. So the fluid and filter which we are showing here in this case has ions which are sodium and bicarbonate. These ions are now in the lumen. Sodium is exchanged for hydrogen. Hydrogen and H plus together, they are going to form what we call H2CO3. Enzyme, carbonic, and hydrase. Now, or sometimes some leave out is that it's non-enzymatic because they can easily recombine together. Now, I want to say something. You can imagine that we are reabsorbing HCO3 minus, see HCO3 minus from here. Let us reabsorb it. We are reabsorbing because it has first of all bounded to H plus to form this structure. This structure will then break down to give you CO2 and more water in the lumen. More water will contain. Now, but CO2 here enters, and we have our CO2, which will combine with our water inside the cells to form what we call H2O. And then H2O together will form H2CO3. This one is where we use carbonic and hydrase. Now, H2CO3 can then divide into two structures. It can divide into HCO3 minus and can divide into H plus. This H plus will still go out again like this. So that's something I want you to note. Then this can also go out here, can diffuse out. So if you see here, we have now entered here and then it is now reabsorbed. So your explanation should be basically what you are seeing from this diagram. That the filtered glomerular, the substance in the glomerular filtration rate is rich in ions like sodium and bicarbonate ion. Sodium is exchanged for hydrogen. Hydrogen comes from a previous reabsorption of HCO3 minus. Hydrogen and the HCO3 minus will form carbonic acid. Carbonic acid dissociates to CO2 and water. Water continues the journey in the lumen, but CO2 is reabsorbed into these cells. CO2 reabsorbed combines with water to form H2CO3 using the enzyme carbonic acid. Then the use carbonic and hydrase. Then carbonic acid dissociates to bicarbonate ion and hydrogen ion. Hydrogen ion goes back again to continue the process with another bicarbonate ion, while HCO3 minus is then reabsorbed. The energy for this step, for this process, comes from the sodium potassium ATPase pump on the basolateral side. That point is quite necessary. And remember to add that this particular phase reabsorbs 80 to 90% of bicarbonate ion, and this occurs in the proximal tube. Now let's go to the second. So that's what you are going to say here for the first one. Now for the second one, which we are going to say we are reabsorbing by primary active transport that occurs in the distal tube. I'll show you here like this, something like this. Then we have our cell. So now you know what this cell looks like. our cell, then our lumen, and then our interstitial side. Now, something interesting happens. So I labeled them against cell, interstitial, and then the lumen. Now, let's do it. Now, if you get to see this, something interesting happens again. But this time, the energy pump is on this side. Same thing like we did before. This time, our hydrogen goes down to join with HCO3 minus. Remember, there was HCO3 minus that was filtered, and that was with sodium. So this one will be exchanged again for this. This one is an AT-based pump. I put ATP. Unlike the other, this is an AT-based pump. Then, this particular structure will join to form H2CO3. This will then give you CO2 and water. This will continue. CO2, just the story we have said before, will bind with water that is inside the lumen, and together we are going to form H2CO3. This will break down to give you H plus and will break down to give you HCO3 minus. HCO3 minus can be exchanged for chlorine. So chlorine can come into the picture to replace it. So that's it again being shown here. The enzyme here was carbonic anhydrous. So what's the difference between this particular one that happens in the distal tubule and the other one that happens in the proximal tubule? The difference was basically that in this one, we are using ATP directly on the apical side. In the other one, we were using ATP from the basolateral side that then was able to couple what was happening at the apical. This one is primary active transport where ATP is used directly. The other one was secondary active transport where ATP was used indirectly. The explanation again is basically what you can see from these diagrams. But also remember to add that these occurs in the proximal tubule and in the distal tubule and accounts for 10 to 20% of reabsorption of HCO3 minus. It's necessary you take that down. Okay? Now let's go down to buffers. For buffers, there are two buffers. The ammonium buffer and the phosphate buffer. The ammonium buffer and the phosphate buffer. Now I'm going to show you now what happens again, the whole story of what I've said. Now inside here, in the sodium buffer here, in the phosphate buffer, I'm going to be showing you that the lumen constitutes of the hydrogen plus sodium H. It constitutes sodium HPO4 minus sodium HPO4 minus, still plus sodium. Now, something happens again. Sodium is exchanged for hydrogen. Now, hydrogen will react with sodium HPO4. And the end result should be that we are going to produce our Na2, then H2PO4. This is what we call the titratable acid. I'll explain each of them in detail. The titratable acid. Now let's explain. What happens in buffering? In buffering, the end result is that we have so much amount of hydrogen. Now, how was that hydrogen? How did the hydrogen come as a result? Hydrogen was as a result of that there was so much increase in H2O plus CO2 binding inside the cell to form H2CO3. Now, the H2CO3 inside this cell can break down to give you HCO3 minus and H plus. Now, if this H plus remains here in excess amount, acidosis becomes even worsened. So we keep losing out this hydrogen. Remember, in acidosis, there is a raised levels of hydrogen. So as we keep losing out this hydrogen, hydrogen goes down from here and it binds with a buffer. What's the buffer we are talking about? The phosphate buffer. On binding with that buffer, we are going to form this particular structure. And this structure that is formed from this reaction is what we call the titratable acid. HCO3 minus, as usual, will be exchanged for chlorine. That is the picture there. Now, what's the explanation again? The explanation is that in the lumen, the body has sodium that has been filtered at the phosphate buffer. Now, inside the cell, water combines with carbon dioxide to form carbonic acid using the enzyme carbonic anhydrase. This is an unstable substance or a stable structure that can easily break down to HCO3 minus and H plus. HCO3 minus can then be exchanged for chlorine. If the levels of hydrogen ions inside the cell increases, this worsens acidosis, and as such, must be exchanged for sodium into the lumen. Hydrogen in the lumen eventually is buffered by the phosphate molecule to form this titratable acid, which is then removed in urine. That's the explanation. I've done that for this particular ammonium buffer, for the phosphate buffer. Now, for the ammonium buffer, and the last here, ammonium buffer is quite funny in the sense that I labeled again the basolateral, the apical, and then the three layers. Now, in the cell here, we can be able to play along with something. We have some amino acids like glutamine. Glutamine constitutes of 60%. Amino acids like alanine and glycine. Alanine and glycine, they constitute about 40%. What do they do? When these amino acids are broken down, amino acids always have an amine group and a keto group. The amine group comes out like this. The amine group is this particular one. While the keto group, I'm going to use HCO3 minus to show you. These can be exchanged for chlorine. Chlorine that fuses and also goes out from here. Now, ammonium here can also diffuse here. And together, both ions can join together to form ammonium chloride. And this ammonium chloride can then be excreted. This ammonium chloride is excreted as titratable acid. So, what have we said in the ammonium buffer? The ammonium buffer is as a result of the breakdown of amino acids, alanine, glycine, glutamine, etc., where alanine contributes to 40% and glutamine contributes to 60%. Now, on breaking down these amino compounds, we are going to yield the amino group and the carbonyl group, that's the keto group side. The keto group side has an active component, that's the bicarbonate ion. So you also see we are indirectly reabsorbing bicarbonate ion here, which also helps to treat acidosis. And for every reabsorption of HCO3 minus, there is an exchange for chlorine. Ammonium here has an active ingredient of hydrogen. And if it remains here for a long time, it worsens acidosis, so must be removed out. Diffusion of chlorine also occurs, and ammonium and chlorine combining together forms ammonium chloride, which is the hallmark in the ammonium buffer, and this is a titratable acid. As simple as that, we are done. So run through these 4 different diagrams and the explanations, and then become a master at it. I must also add again that in buffering, another essay can be added right on blood buffers, or another essay can be added right on acid-base balance. Acid-base balance in particular, if they say you write on the major buffers in acid-base balance, it indirectly means you write on ammonia and phosphate. But if they ask you for the acidification of urine indirectly also means that you should talk on buffering. That is the ammonia and the phosphate. Now, to end this here, I will talk about respiratory response and alkalosis. For response and alkalosis, number 1 is that condition that makes this possible is that there is a fall in hydrogen ions, and this will prevent the reabsorption of HCO3 minus. That's the first point you should make as a condition, because whenever we want to reabsorb HCO3 minus, we need hydrogen ion. So in people with alkalosis where there is a fall in H plus, it becomes difficult to reabsorb HCO3 minus. Now, the mechanism for response in alkalosis will be that there is going to be an increase. There's going to be an increase in the loss of HCO3 minus in urine. This persists for a long time. This will persist for a long time until homeostatic levels are reached. Until homeostatic levels are reached. So, this is an essay on acid-base balance, and I really do hope it made the most sense to you. If you need a prototype or an already written essay on acid-base balance, you can also endeavor to comment there on the comment section. I'll pin out a number there where you can send a message, and I'll personally send you these particular essays to you. Endeavor to like this video, endeavor to share these videos to your friends, subscribe to this channel, and follow for more of these videos. Until then, see you next time.